Last time I talked to you about mitosis, the division of a cell to make two identical copies, but I wanted to talk to you about a totally different type of cell division today, and this is a type of division that's called meiosis. Meiosis is really similar, yet really different at the same time. And I know it's super frustrating, but for some reason, mitosis and meiosis got names that are really super similar. But remember, I talked about how mitosis was the action of threads, the movement of DNA in the cell. Let's take a look at what we mean by meiosis. So, meiosis you can actually break down just like we broke down the term mitosis. Osis, remember, means action. And so we're talking about some kind of action, some kind of process that's going to happen inside of the cell. But meio has its own meaning. So meio means to lessen. I know that probably doesn't make sense right now, but it will definitely make sense by the time I've done this. You see, what we're going to lessen is the amount of DNA in a cell, because meiosis is all about the creation of cells that have 1/2 of the DNA of a standard cell. That's right, meiosis is used to make sex cells, or sperm and eggs. It is used to make what we would call gametes. Then that's exactly what we mean by gametes, is cells with 1/2 of the DNA of a standard cell. So when we're talking about meiosis, we're actually talking about cell division that makes gametes. And when I say gametes, what I mean are cells with 1/2 of the DNA. So there are different kinds of cells in our body because there's different kinds of functions that need to be performed, but almost every cell I am made out of is called a somatic cell, and it's a cell that really is just like performing some function in my body. But every organism that is going to sexually reproduce, that means have two parents that combine genetic material, they are going to produce what are called gametes. And gametes are these cells with 1/2 of the DNA. So why does an organism need gametes? Well, it all comes down to sexual reproduction. During the process of sexual reproduction, you have to have two parents. And sexual reproduction is going to combine genetic material or combine DNA from two parents to make genetically unique offspring. Typically, what's going to happen during this process is there is going to be a sperm. And this sperm carries 1/2 of the DNA of a standard cell. That sperm is going to combine with an egg. Now, this is not to scale. The egg would actually be way, way bigger than the sperm. The sperm would be much, much tinier than that. But it also has 1/2 of the DNA. When you add those together, you are going to create a new type of cell called a zygote. And that zygote is going to have 1/2 plus 1/2, well, one full amount of DNA. So what this means for humans is we have 46 chromosomes in all of our somatic cells, but we have 23 chromosomes in our gametes. This way, when a male sperm combines with a female egg to make a human zygote, that zygote carries 46 chromosomes. If something doesn't carry 46 chromosomes, that can lead to abnormalities in the way that the organism develops. You see, having a different amount of DNA changes the way that the cell functions because it either has too much or too little information. And that changes the proteins that are being made and the ratios of those proteins. That ultimately changes the function of the cell. And so that's what meiosis really is it's trying to get a cell that has less DNA. And so the name, an action that lessens, hopefully makes sense because it's an action that is going to lessen the amount of DNA in the cell. So how does this happen? Well, as you probably guessed, it's going to happen in phases, just like mitosis did, but it's going to take a lot more phases. So meiosis actually has more phases than mitosis, and it actually ends up having two separate times where the cell is going to split. So we often end up talking about meiosis I and meiosis II, each of those having its own phases. So let's start with just the simplest of the phases, and that would be meiosis I, in which we have prophase I. So prophase I is going to be very similar to prophase in mitosis in the sense that we're going to have our cell, and our cell is going to go through kind of the same changes. There would initially have been a nucleus. There would have been strands of DNA. Those strands would have already duplicated through the S phase. The nucleus would dissolve, and the strands of DNA would form into chromosomes. Now, this is going to lead to something a little bit different than what we saw happen when we dealt with mitosis. So I just need to add some chromosomes, and I'm going to color code these chromosomes because the chromosomes do something a little bit different this time. Do you remember when we talked about the types of chromosomes and how humans have 46, but they really count as like 23 pairs because we have duplicates? Well, those duplicates that we got from your mother and your father are called homologous. And so when we talk about homologous chromosomes, we're talking about ones that kind of code for the same information, but maybe have different alleles. What's going to take place in prophase I is the homologous chromosomes are going to find each other and pair up. So I had drawn my chromosomes like this, but let's pretend that the reds are chromosome number 1, and let's pretend that the greens are chromosome number 2, and let's pretend this organism only has 4 strands of DNA inside of all of its cells. So what would end up happening is the 2 red chromosomes are homologous, coding for the same information, but possibly having different alleles or different versions of that DNA strand. They're actually going to find each other and they are going to pair up together. The greens are going to be homologous chromosomes as well. So at some point in this process, they are going to find each other and pair up. Now, there is another weird thing that happens here is that homologous chromosomes actually cross over, meaning that they swap pieces of DNA with each other, which is like super weird and it happens, but at this level we don't really have to understand it. But that is another thing that takes place is homologous chromosomes will cross over, and that creates even more genetic diversity. And that's kind of one of the points of meiosis is to increase the genetic variety within a species. So having a whole bunch of different organisms with different DNA. Remember, one of the things we talked about with evolution is that variety or diversity is one of the most important things in a population and can help lead to natural selection and ultimately evolution. And so meiosis is set up with a lot of mechanisms that help ensure the increase of variety and diversity within the genetics of a population. So this is our process of prophase I. You see, prophase I is able to send the DNA so that it creates those chromosomes, you lose the nucleus, they pair up into homologous pairs, and then we move on to our next phase. So the next phase that's going to take place, if you probably guessed already, if we had prophase, we're going to have metaphase. And metaphase is going to look very, very similar to what we saw in mitosis. The only difference is, remember, we have homologous pairs of chromosomes. So when we take a look at what's happening during meiosis I, metaphase I, we have our cell, and within that cell, we have our homologous pairs. And those homologous pairs are going to line up in the middle. Meta literally means beside. So this is the phase where chromosomes end up beside each other. So they line up down the center of the cell. So I have my one set of homologous chromosomes. I have my other set of homologous chromosomes. And they are lined up right down the middle of the cell. Now, the other thing that would end up happening is you would also have spindle fibers. So I'm going to draw those in blue this time, and they would attach to each of the homologous pairs, again at the centromere, the middle of the chromosome. So that is all that takes place within metaphase I. And after metaphase I, just like in mitosis, we're going to lead to anaphase, but this is going to be anaphase I. So I left up my drawing of metaphase I so that I could explain what's going to happen next. You remember we had our homologous chromosomes that were lined up next to each other, and I'm going to deal with the red ones very first. What actually happens in anaphase is, rather than splitting the actual chromosome itself apart, anaphase I pulls the homologous chromosomes apart. So you end up getting a homologous chromosome on one side and a homologous chromosome on the other. This would then again happen with the other homologous chromosomes, where one gets pulled to one side and one gets pulled to the other. So now we have a homologous green and a homologous green on opposite sides. This is super important. Remember, you actually have to get the right kinds of DNA spread across these gametes so that when they combine, you actually bring in one homologous chromosome from both the mother and the father. So that means you have to get a chromosome number 1 in a sperm and a chromosome number 1 in an egg, and a chromosome number 2 in a sperm and an egg, 3 in a sperm and egg, all the way, if we're talking humans, up to the 23rd chromosome. So you actually have to make sure that you're sending the correct chromosomes to the gametes. There's also another little trick here, and that is that in meiosis, when those homologous pairs line up, they don't necessarily always line up with the mother on one side and the father on the other. It's kind of randomized, and that again increases the genetic diversity, because now all of a sudden there's a huge amount of different ways that you can have those sperm and eggs carrying DNA. They can all have genetically unique sperm or eggs, and that means when they combine, there's a huge amount of variety that could be created from those sperm and eggs. So that is anaphase I, which leads us to telophase I. So during telophase I, we had those chromosomes, the homologous pairs, being split apart, and now we are going to end up with two separate cells. Inside of each of these cells is going to be those chromosomes. So we have one of the reds on each side, and then one of the greens on each side. Now, it's probably also important to understand at this point, what we have created is two cells that have duplicated DNA, but even though it's duplicated, it actually isn't really enough for this cell to operate as a somatic cell. This cell actually doesn't really have enough information in it because it's carrying, let's say this is a human chromosome, it would technically have 46 strands of DNA, but 23 of those are exact matches for the other strands. So you have duplicated DNA, but really only half of what is supposed to be in a standard cell. And so now we have two cells, but they still have that duplicated DNA, so we need to divide again. And that second division and the process of it is called meiosis II, and it's the second set of phases. It's going to go through the exact same sets of phases, prophase, metaphase, anaphase, and telophase, until it can divide again. So let's take a look at what happens next. So now we're dealing with meiosis II, and really the process of prophase II. And so that is now what we have moved on to. And it's important to understand that there's actually two cells going through each of these steps. And so prophase II is really just kind of like a resting phase. Now, it's important to recognize that some organisms actually take a very long time to transition between meiosis I and meiosis II. So in human females, for example, meiosis I ends during the menstrual cycle at a point when it's about to release an egg, but meiosis II doesn't really take place until possibly that egg is about to be fertilized. So there can be a big gap between meiosis I and meiosis II. So you may actually have the reformation of a nucleus, the unfurling of the chromosomes, and so you do need prophase II in order to deal with that. So we have our prophase II, we have our chromosomes, they're hanging out inside of our two cells here, and this is going to lead us to our next phase. So just like before, we had prophase I, we went to metaphase I, here we're going to go prophase II to metaphase II, and as you probably guessed, metaphase, middle. Chromosomes are going to line up in the middle of the cell. So during metaphase II, I have my two different cells that are going through this. I'm going to have my red chromosomes. They're going to be lined up in the center with my green chromosomes. And then the next thing that's going to happen, that always happens during metaphase, is our spindle fibers are going to attach. So spindle fibers attach to the center of each of those chromosomes. And that's all there is. That leads us to our next phase. So after metaphase II, we move on to anaphase II, and now we have these duplicated chromosomes. Now is the point where they're actually going to be pulled apart from each other. So now during anaphase II, we're going to have the division of chromosomes to opposite sides of each of those cells. So now we've reached anaphase II. Here's what we're going to do next. Those chromosomes are now being pulled apart from each other. And so you end up getting one strand of DNA moving to opposite sides of the cell, and this happens for each of those cells. So greens and reds getting pulled apart and moving to opposite sides of the cell. Spindle fibers are the things that are actually doing this. So we have our spindle fibers still attached, providing that like rail network in order to actually allow these chromosomes to move. Okay, so we're almost at the end. We've finished anaphase II, we've divided those DNA chromosomes across to opposite sides of each of those cells, and now it leads to the last phase of meiosis, which is telophase II. So in telophase II, we're going to end up where those two cells that we used to have are now going to divide again. And this is kind of the whole key to meiosis. Meiosis is going to create 4 cells. Each of those cells is going to have half of the original DNA of a standard cell, and that is going to create what we would call a gamete. So each of these cells is a gamete. Gametes are called haploid cells because they have half of the DNA of a standard cell. So this is what we end up creating, is these gametes. And gametes would be things like sperm and eggs. Gametes are something that exist in all sexually reproducing organisms. So whether it be animals, whether it's like humans or chimpanzees or octopi or any sort of animal that sexually reproduces, but it also happens in plants. And so plants are able to sexually reproduce, and they're able to use gametes in order to help them create genetically diverse offspring. So again, meiosis is also this incredibly old evolutionary trait that has been carried through the history of life because it is so successful at creating genetic diversity. And it does that through the pairing of homologous chromosomes to allow them to make sure that gametes have the proper DNA, but also doing it in a way that increases variety by recombining and having crossing over, by allowing homologous chromosomes to be on either side, and ending up creating all of these genetically unique things by combining two sets of DNA together from two parents. And so that's the process of meiosis. And I hope this gives you a better sense of what this process actually is and how it is so incredibly different yet similar to mitosis. So thanks again for watching, and I'll see you next time.